Today we're on Chapter 10 of the Chemistry 2 textbook, Biological Polymers. This is the last chapter in the textbook, and we want to talk about several things. We want to talk about polymers in living things. We want to talk about proteins, which are a type of polymer, and look at some of the structure of proteins, and then talk about nucleic acids and the idea of the genetic code. Talk about RNA, an important part of the protein synthesis process, and move on from there. Now, the cell is a complex structure, very complicated. It carries out a lot of functions. The cell makes molecules, it changes molecules, it uses molecules, it disposes of molecules. Anything you can do with a molecule, the cell does. Many complicated structures are needed to make these things happen. These structures that accomplish these things are very complex, very highly organized. And you need an overall control processor to regulate cell activities. You can't just have parts of the cell going wild, doing whatever they feel like. You need something to control all of it. Now, there are two major polymer types in the body. The first one is proteins, and proteins are composed of amino acids, and we'll talk about protein structure in just a minute. Some of the types of proteins are enzymes, which catalyze chemical reactions in the body, transport proteins, which move things from one place to another, structural proteins, which, as the name implies, provide structure to the body, such as proteins in bone, and some hormones. Insulin, for example, is a protein hormone that regulates certain processes. DNA and RNA are composed of nucleic acid chains, and these polymers are responsible for protein synthesis, and we'll see more about that later. Now, here's a couple of examples of proteins. The DNA polymerase on the left is an enzyme. It copies DNA and helps make new proteins. The kinesin on the right is a transport protein. It moves materials from one place to another in the cell. It's a phenomenal little device. It's just amazing how it works. Now, let's look at protein structure for a second. Proteins are composed of amino acids, and we'll define an amino acid momentarily. Now, we can talk about protein structure at four different levels. The first level of structure is the primary structure, which is basically the amino acid sequence. How are the amino acids arranged in a chain in the protein molecule? Secondary structure is local folding, and we'll illustrate that, but there are segments of the protein that might fold or bend a certain way. The tertiary structure is the overall three-dimensional shape of the protein. What does it look like in 3D? Sometimes a protein may have quaternary structure if that protein consists of more than one subunit, and we'll illustrate that. Now, the primary structure is made up of amino acids, and amino acids have a carboxylic acid group and have an amino group, a central carbon, and an R group that can be anything from a hydrogen up to a very large molecule. The amino acids link together to form a peptide bond as they form an amide bond. We talked about amide linkages in an earlier chapter, and that's basically what that is. Now, here's an example of an amino acid structure, the amino acid leucine. You see the carboxylic acid group, the central carbon, the amine group, and a particular specific R group, which defines that amino acid as leucine. And it's abbreviated either LEU or people are starting to use the one-letter abbreviations nowadays, and that would be abbreviated as an L. The amino acids form peptide bonds. The carboxylic acid group of one amino acid joins with the amino group of another amino acid to form the amide linkage, the C double bond O NH linkage that you see on the right hand of the screen. In proteins we call it a peptide bond, but in organic chemistry we call it an amide bond. It's exactly the same thing. Now the secondary structure is the local folding, and we have two major types of secondary structure. The alpha helix over on the left in the purple protein, you notice that's coiled, and that forms the alpha helix. The beta sheet, or sometimes referred to as the beta pleated sheet, is kind of a wrinkled corrugated cardboard looking type of a pattern, and sometimes you'll find the yellow arrows drawn onto a protein structure to indicate that. And then there's other types of the secondary structure that are just random folds. They don't have a pattern, so we don't bother naming them. Tertiary structure is the three-dimensional picture of the protein. And on the left we have a model of the DNA polymerase, and you can see the different pieces of that, and you can see how it looks in 3D. And you can also see marked alpha helix portions, beta pleated sheet portions, and different parts of the protein to indicate what it does. There's some other examples of tertiary structure. These are just stray proteins that I picked up off the internet. But again, you can see where the alpha helix and the beta pleated sheet are both clearly indicated on the protein to give you an idea of the structure. Quaternary structure, if a protein has more than one unit in it, it has quaternary structure. And there's a picture of a proteasome, which is a protein that breaks down other proteins. And you'll notice that the quaternary structure of this is made up of several smaller protein units. Another protein that we're familiar with is hemoglobin. Hemoglobin is involved in the transport of oxygen in the bloodstream, and hemoglobin is made up of 4 subunits. The blue structure on the left is a globin monomer. 4 of these go together, as indicated, to form the hemoglobin molecule. The red and the blue stand for 2 slightly different types of structures in the hemoglobin. Now, nucleic acid polymers are important. They're composed of a nucleic acid base plus a phosphate group plus a carbohydrate. A little more complicated in terms of the structure. The DNA, deoxyribonucleic acid, is the molecule that contains the pattern for the RNA structure, and as we'll see, that determines the protein structure. So in essence, the DNA is the blueprint for the structure of a protein. RNA contains the direct pattern for the protein structure as produced by the DNA. Other RNA molecules are important. The transfer RNA is a small RNA molecule, and that's involved in guiding the specific amino acids into the growing protein chain. Ribosomal RNA is a large molecule, and ribosomal RNA is involved in the actual assembly of the protein in the ribosome. That actually helps put the amino acids together to form those amide linkages. Now, here's a picture of the DNA structure. We have a nucleotide connected, a hydroxyl group of one nucleotide connected to a phosphate group on another nucleotide, and you develop a long chain. The DNA sequence is important. You have 4 different bases in DNA, and the sequence in which they're rearranged is very important because that's the blueprint for the amino acid. A 3 nucleotide structure, say ATC in this particular molecule from top to down to, would specify a particular amino acid. So if we had ACT, that would mean a different amino acid. So the arrangement of nucleotides is pretty important. The DNA forms as a double helix. And here's a picture of the double helix, the double strands, and the backbone. Now, RNA is a single-stranded molecule, but it's very similar in structure. It has the phosphate group and the oxyribose nucleic acid. So it's very similar. It's just a single strand. Transfer RNA is a small RNA molecule, and it has kind of a unique shape. And you notice the molecule on the left, up at the top, is where the amino acid would attach. And then down at the bottom is a 3 nucleotide sequence known as the anticodon, and that's where the transfer RNA links up with the regular RNA to put this particular amino acid in the right space in the sequence as the protein chain grows. Now, when we make a new protein, transcription is the first step. An RNA polymerase binds to DNA and copies the DNA. So the RNA polymerase makes an RNA molecule according to the pattern of the DNA so that the right protein can be made. Then translation takes place. The messenger RNA attaches to the ribosome, which is a very large molecule. The transfer RNAs bring in the amino acids and line up with the RNA. Each anticodon in a transfer RNA matches a codon in the RNA so that the transfer RNA can get into the right place. And then the ribosome connects the amino acids by forming the amide linkages to build the protein. Here's a summary of the whole process. In step 1, there's the DNA. Step 2, the DNA strands separate and you make an RNA molecule. In step 3, the RNA links up with the transfer RNA amino acid complex, and in step 4, the ribosome forms the amide linkages for the growing protein. Okay, we've talked about protein structure, the importance of protein structure, and how we describe protein structure. We've talked about DNA structure, DNA and RNA. We've mentioned the genetic code, haven't gone into a lot of detail about it, but we have given a brief description of the genetic code. Then we talked about the roles of RNA and transfer RNA in putting the amino acids together to develop the protein. And we looked at an overview of the process of protein synthesis. It's an amazingly complex and complicated process, and we just kind of hit the highlight. So, thanks for being a part of the class. This ends the Chemistry 2 series, and I enjoyed having you in the class and hope everything goes well with you.